Hello, everyone. I'd like to welcome you to the Genetic Code Expansion webinar series. If you are here, you know that this happens every third Thursday of the month at the same time, which depends on whatever time zone you're joining us from. And if you're here, don't worry about when the next one will be, because we'll send out reminders for when it's going to happen. To point out that this webinar is important for keeping the field up to date on the latest and greatest in genetic code expansion. It's important we want it to be important for education and also to bring in fresh ideas. Another important aspect is that we would like to give early career scientists an opportunity to share their work in the lab that they have just started, or if there's a postdoc looking for job opportunities that wants to share their work with us, we'd ask for you to send in names of individuals that you'd like to hear from, or if you would like to share your work with us, please email us and we'll try to get you on the docket to present. I'd also like to point out that there's a genetic code expansion workshop starting August 2nd through the 8th, which will have hands-on training focused on selecting your synthetases of interest, which will be followed by an exciting conference in genetic code expansion, all taking place in Corvallis, Oregon. So please look into this if you're interested. We have it shown here in the next couple seconds. If you want to scan the QR code, you can get all the information for it. All right, so we have two great speakers today. And what I really appreciate about both of them is that they probably refer to the surface membrane of the cell as the membrane. We have Dr. Isabella Derler from Johannes Kepler University and Dr. Tom Sackmar from Rockefeller University. To start off, we have Dr. Isabella Derler. She was trained as a biophysicist by Christoph Romanin at Johannes Kepler University, where she studied calcium signaling and calcium ion channels. She then went on to train with Mitsu Akura in Toronto as a postdoc to in 2008, investigate the emerging fields of crack channels and specifically Orai1 and its regulation by lipids, which is extremely exciting to me, to overcome hurdles of transient interactions and channel components that are crucial for drug discovery. She started down the path of genetic code expansion. And this is about the same time in 2019 where she was appointed as an assistant professor at Johannes Kepler University, where she studies an emphasis on biophysical signaling. Her lab is using all sorts of cool biophysical approaches, including electrophysiology, fluorescence microscopy, biochemistry, and molecular dynamic simulations. And today she'll be telling us about novel molecular insights into the Orai1 channel dynamics using photocrosslinking. Take it away, Isabella.
Okay, so thank you very much for the introduction and welcome to my presentation. And thank you also for organizing this great webinar. So in my talk, I would like to provide you insight on the one hand, how we used photocrosslinking unnatural amino acids to transfer light sensitivity to a calcium ion channel Orai1. And on the other hand, how we got some novel insights in the mechanisms of the Orai channel pore opening. So as you have heard, we are working on calcium ion channels, and our main interest are calcium ions in the human body. And you know that calcium ions play a role in bone growth, in muscle contraction, heart function, fertilization, in immune response, and neuronal signaling. And all these processes are regulated at the level of single cells, where the exchange of calcium ions across the membrane and across intracellular organelles. And we are in particular interested in one calcium ion channel, which is activated by the depletion of intracellular calcium stores. It's the calcium release activated calcium channel, also called crack channel. And it's activated by the binding of a ligand to a receptor at the membrane, which initiates a signaling cascade within the cell, which finally leads then to calcium store depletion from the largest intracellular calcium store, the endoplasmatic reticulum. And this finally leads to calcium influx from outside into the cell. And this linkage between calcium store depletion and calcium entry is known now for more than 50 years. And the two molecular components which are essential for this linkage have been identified a bit more than 15 years ago. And one of these two linker proteins is the STIM protein. It's a calcium sensor protein located in the endoplasmatic reticulum in the membrane. It can sense the calcium concentration in the lumen of the endoplasmatic reticulum via the CF hand. And the second important protein is a calcium ion channel, Orai1, in the plasma membrane. And mechanistically, it works like this. that in the resting state, the STIM protein is located tightly packed in the membrane of the ER with calcium bound. And upon store depletion, it loses the bound calcium, it undergoes conformational change, reaches to the plasma membrane, and can bind to the Orai channel to allow calcium to enter. And this is the so-called calcium release activated channel, and the calcium entry via this channel is in part important for inflammatory mediators in T cells, but it's also important in B cells, in mast cells, and apart from the immune system, it's also important for muscle cells, the endothelium, platelets, and melanocytes. So overall, it's quite universal. And since the discovery of the two proteins, STIM and Orai, a variety of defects in both proteins have been identified, which can lead to a variety of diseases. And it can be said that overall said, when a downregulation of the function of STIM or Orai occurs, this can lead to severe combined immune deficiency, autoimmune disease, or muscular hypotonia. An upregulation of STIM or Orai function can lead to cancer, tubuloaggregate myopathy, where patients suffer from muscle weakness, cramps, or contractures, or syndromes such as the Storm-Walker syndrome or the York platelet syndrome, where patients suffer from a variety of dysfunctions in the body as displayed here. And for us, it's important to provide a detailed understanding on the mechanisms of these proteins. And currently, we are focusing extensively on the function of the Orai1 channel. So there are already structures available of this channel. And you see here, it has this beautiful hexameric symmetry, which is unique among calcium ion channels. So most calcium ion channels have a tetrameric symmetry. And each of those subunits is composed of 4 transmembrane domains. And an important feature of this channel is, on the one hand, the calcium ion pore in the center, formed by 6 team-1 domains. And another important property of the channel is the periphery of the channel complex, where you see here the C-terminus, and always 2 C-termini of adjacent subunits undergo this antiparallel conformation. And it has been shown that the C-terminus is the main coupling site for STIM. So when STIM binds there, then pore opening happens. And it is assumed that when STIM binds here, a conformational change is initiated, which propagates finally to the pore to allow calcium entry. But the conformational changes which occur have not been understood in detail so far. I mean, there are structures available, and we did a lot of site-directed mutagenesis. So I would like to show you here what we performed here to understand the pore opening mechanism using canonical site-directed mutagenesis. So typically what we are using is the whole cell patch clamp technique, and the whole cell patch clamp technique, and we are investigating cells containing STIM and Orai overexpressed. When we attach the pipette to the cell and these 2 proteins are typically in the resting state. And after applying the stimulus, to induce the store depletion, STIM and Orai get activated, and we see in the time course after some time then the activation to a maximum. But when we incorporate, for instance, in the team-1 domain a gain of function, a mutation, it can lead to gain of function. So what you see here, you have already activation with whole cell break-in, independent of store depletion and independent of STIM. And with time, we found a set of different mutations in all the different 4 TM domains. And overall, there are approximately 13 to 15 positions, which when mutated can lead to gain of function, as shown here. Some of these are also disease relevant, as here in these red rectangles. And as not only in the pore lining TM domain, but also in the non-pore lining TM domains, we can get gain of function with site-directed mutagenesis, in particular also from the periphery of the channel complex. The conclusion came up that Orai pore opening requires a global conformational change of the entire channel complex. There are several structures available. So there is a crystal structure of the closed state. There's a crystal structure of the open state of a gain of function mutant, but in the absence of the physiological activator. And you see the main difference is here, that here the part at the periphery, so team 4 and the C-terminus are kinked and bent, and here it's fully straightened. So it's assumed that such a conformational change is associated with pore opening. Another study showed that each subunit undergoes rigid body outward movement, so this direction, to induce pore opening and to induce widening of the pore. And then there are also MD simulations available, which also showed that there might be a twist to open gating mechanism involving also rotation of the TM domains. So a wealth of knowledge is available. It's consistent that we have a global conformational change, but there are also some inconsistencies at the moment, and all the structural studies have been performed in the absence of STIM and also provide only a static view of the channel complexes. And what we thought might be interesting to monitor inter-TM domain motions. And for that, we came across the genetic code expansion technology. And we thought that maybe the photocrosslinking unnatural amino acids could help to get a better idea on individual TM domain motions and the dynamics of the channel complex. And yeah, as you all know, so the photocrosslinking unnatural amino acids undergo a covalent bond formation upon UV light irradiation in the range of 3 to 4 angstrom with a residue in close proximity. And we focused here in particular on the acetophenylalanine and the benzophenylalanine. And we thought if we incorporate one of these unnatural amino acids at different positions in the TM domains, we could trigger with UV light a local conformational change, which could then be transferred to the entire channel complex to lead to a global conformational change. And this is what we tried. So initially we searched for positions in the Orai channel which could be suitable for UAA incorporation. And we thought, so we went back to our screen and we thought that these positions which already lead upon canonical substitutions to gain a function could be also suitable for incorporation of AC and BPA and be sufficiently flexible to lead to conformational changes. So we used these positions, we incorporated at these positions in team 2, team 3, and team 4, the acetophenylalanine, or short AC, or the benzophenylalanine, here short the BPA. And we were quite lucky that we saw in all these mutants a very nice plasma membrane localization of the Orai channel, which is typical for the Orai channel. So we could incorporate at all these positions the unnatural amino acid without affecting the localization of the channel. And we investigated then these individual mutants with calcium imaging using RGECO as a calcium indicator. And we find, so as a comparison here, you see the STIM Orai containing cells and their calcium levels before store depletion and after store depletion, there strongly increases. And then here all the mutants before UV with the filled bar and after UV with the hatched bar. So you see here for some positions functional changes, and we focused on 3 positions in more detail. For all these you see there is low or no activity or no calcium entry before UV light, but it strongly enhances when you apply UV light. So we have identified mutants which allow UV-induced activation, and to confirm this, we further performed calcium image, performed further patch clamp experiments using the whole cell mode. And to remind you, so we attach the pipette to the cell containing STIM and Orai. In the beginning, the STIM and Orai are in the inactive state, and only upon perfusion of EGTA, a calcium chelator, which triggers then passive store depletion, we can get the activation of the STIM-Orai complexes, and this is seen here in this typical STIM-Orai time course. So you get, after some time, the store-operated activation. If there is only Orai1 in the cell, this is not visible, and also UV light cannot affect the Orai functionality. And if you look at the current voltage relationship here, you see it shows a nicely inward rectifying current with a reversal potential in the range of plus 50 millivolt, which is typical for these cracked channels or STIM Orai channels. So in comparison now, the 3 mutants I picked out before, they show all no activity before UV light, but after UV light, they activate within 15 seconds to a maximum. And if you look at the current voltage relationship, you see it's identical to that what we have in the presence of STIM Orai currents, also with a similar reversal potential which led us to conclude that we have Orai mutants which lead to cracked channel-like currents already in the absence of STIM. So the incorporation of the photocrossing UAA allows cracked channel-like activation. As I said, it's in the absence of STIM. To prove this further, we mutated the main binding site and the C-terminus of Orai1. So it's this mutation. We incorporated this mutation in the UAA containing mutants, again, these three. And we still see the UV-induced activation, so proving that STIM1 binding is not required for the UV-induced activation. We further performed experiments in CRISPR-Cas9 co cells. So if endogenous STIM1 Orai1 is missing, or STIM1, STIM2 is missing, or all the three Orai isoforms are missing, we still get the UV-induced activation for all the mutants. So photocrosslinking-induced Orai1 activation occurs independent of STIM1. And it's known that Orai1 is quite important in immune response, in particular in T cells. Their calcium entry triggers NFET translocation to the nucleus to induce gene transcription. And in the next step, we wanted to identify whether the UV-induced Orai1 activation is also suitable to trigger NFET translocation. And you see here a cell which contains this mutant, so A137BPA, and it's co-expressed with NFET in the cytosol. And it's nicely here in the cytosol. And when you apply before UV light, and if you apply then UV light, it goes to the nucleus. Also seen here in the diagram, before UV light compared to after UV light. So after UV light, it increases the NFET translocation within 30 minutes to a maximum, which is not occurring for the mutant in the absence of UV light and also not for wild-type Orai1. So overall, photocrossing-induced calcium entry is suitable to trigger downstream signaling. So UV-induced Orai1 activation occurs in the absence of STIM, but we still wanted now to identify if STIM can still activate these mutants or whether the UAA affects somehow STIM activation. You see here again the three mutants, and in all three cases in the presence of STIM, we can see this typical store-operated activation as we have it already seen for the STIM Orai currents. And if you then apply the UV light, we still get a bit further activation. So photocrosslinking UAAs do not interfere with the evolutionary defined activation pathway, but we found some interesting feature for this mutant. So in comparison, when we have first STIM activation and then UV light, if we apply first UV light, then we get larger currents, which do not further enhance when STIM1 couples afterwards. So we get larger currents with first applying UV light, while when we have first STIM coupling and then UV light application, currents are smaller. So which indicates that STIM binding somehow interferes with maximal photocrosslinking-induced conformational changes or photocrosslinking-induced activation. And we are currently pursuing this a bit more because at the moment it's clearly known the C-terminus is important for binding, but not much is known about direct binding to the loop 2 or the N-terminus. So this might be a hint that STIM is also somewhat directly affecting the area here, and we are trying to find out this now more. Another interesting feature has this mutant. So we go back to the mutant in the absence of STIM. And what you see, or what you have already seen before, it shows this nice UV-induced activation and the typical current voltage relationship. And we looked and in the structure, where is it located? And what you see here is that the A to 5 at the periphery, so in TM4, so at the periphery of the channel complex, it's somehow directed to TM3. If you look from the top view on it, you see here A to 5,4, it's directed to these two residues. So we cross-checked again with our screen we performed before. And there we found that in particular this position shows also upon incorporation of photocross-linking unnatural amino acids, UV-induced activation. We confirmed this with patch client experiments, and what you see here is V181 or BPA, both showing UV-induced activation with a typical current voltage relationship of cracked channels. It's not as high like for this mutant, But anyway, for 2 positions which are opposite between team 3 and team 4, we get photocrosslinking induced activation from both sides. And this somehow indicates also a communication between team 3 and team 4. And to understand this communication in more detail, we continued with double site-directed mutagenesis. So on the one hand, we incorporated at V181 ATSI or BPA. And on the other side, in team 4 at 8254, we incorporated different canonical amino acids with increasing side chain size. So we have the glycine, alanine, cysteine till tryptophan. And you see here, we get modulation in the current levels for these double mutants. But if you look here, if you look at the current over the amino acid volume or the calcium level over the amino acid volume or size, you will see there's a tendency for increasing current before, but in particular also after photocross-linking. The same for the BPA mutant. So overall, this indicates that with increasing side chain size at this position, we get increased photocross-linking induced currents. This further indicates that likely if we increase the distance between team 3 and team 4, we get more calcium entry. And to prove this further, we performed MD simulations. We looked on MD simulations of the closed state, and there we found that the distance between team 3 and team 4 is in the range of 6 angstrom. And we thought we could restrain the distance between team 3 and team 4 at 8 angstrom, 10 angstrom, or 13 angstrom. And what we found is with increasing distance, we get increasing hydration of the pore and also increased dilation of the pore. Further, we also found some structural rearrangement in the channel complex, while in the closed state, we have more interactions between team 1 and team 2. In the open state, we get a rearrangement to more interactions between team 1 and team 3. So our restraining the distance leads to, again, a global conformational channel change in the channel complex and triggers pore opening and enhanced hydration. So we think that pore opening really requires a dilation between team 3 and team 4. And so far, these studies were all done in the absence of stim. So we continued a bit also in the presence of stim with these mutants. So we co-expressed our UAA-containing mutants with stim. And what we find here is that this is, I'm showing here as an example, the V181 ATSI mutant. In the absence of stim, you have the UV-induced activation. In the presence of stim, you see first store-operated activation because of stim. And when you then apply UV light, we get strongly enhanced currents, which are larger than in the absence of stim. So we asked us now, what is happening now? So stim is binding, and it triggers larger currents, meaning it must trigger larger conformational changes which somehow facilitate the photocross-linking induced activation or improve photocross-linking induced activation. So we thought, what happens if we do photocross-linking before STIM1 induced activation? So we did this. So we did first the photocross-linking here, likely between team 3 and team 4. You see here again the trace with first stim binding and then UV-induced activation. But when we do it the other way around, first UV light application, we get the UV-induced activation as we see it also in the absence of stim. And then we get a bit further activation, likely due to stim coupling. But it does not reach the level like we have it when we have first stim coupling and then UV-induced activation, which indicates that with cross-linking, we hinder somehow the STIM1 induced activation, maximal activation. And we believe at the moment that these results somehow indicate that STIM1 binding triggers a conformational change, likely between team 3, team 4 interface to induce pore opening. Yeah, here if I'm at the end of my talk, and in conclusion, we can say that incorporation of photocross-linking unnatural amino acids in TM domains was successful. We could really trigger UV-induced calcium influx, we could trigger downstream signaling, and we could get some novel mechanistic insights in the pore opening mechanism of the channel complex. So on the one hand, we found that the extent of photocross-linking induced activation correlates with the side chain size along the TM3, TM4 interface. On the other hand, we could also show that the widening of the TM3, TM4 interface is likely a physiological conformational change induced by STIM1. And due to distance enhancement, we really got also this global conformational change in the channel complex. Yeah, and in the end, I would like to thank my entire group in Linz and the ion channel group also in Linz for their contributions to these nice stories, our collaboration partners, and thank you for your attention.
Thank you very much, Isabella, for the great talk. If there's any questions, you can put it in the chat and I can ask it. I'll start off. So now that you have the ability to really effectively use genetic code expansion in Orai, and being that it's a 6-member channel, have you looked into looking at stoichiometry to see like how many of these cross-linking events need to occur for activation?
Yeah, we tried in this direction. So we did a co-expression of wild-type Orai1 and the Orai UAA containing mutant, and we found that with increasing Orai1 wild-type, we get less UV-induced activation. So we really think that at least 5 or 6 UAAs have to be in the channel complex to allow current, so UV-induced current enhancements.
You did it in like a mixing experiment, basically.
Yeah, we did a mixing experiment.
Yeah, very cool. And then maybe a follow-up, if you tried the really difficult thing of now following up with looking at doing the mass spec to see where these cross
yeah, the first step would be the purification or some of the Orai channel, and I think many people are struggling with this. So we are thinking of it, but we did not successfully accomplish with this.
Yeah, it's the bane of the cross-linking community, right? Is when a reviewer asks to look for where it cross-links to.
Exactly.
But yeah, all right, thank you. Are there any other questions? Looks like hold on one second. I'm looking for it. This is from our next speaker, Tom Sakmar. Is it counterintuitive that cross-linking should induce rather than inhibit the current?
Can you repeat the question again?
Yeah. Is it counterintuitive that the cross-linking should induce rather than inhibit the current? So can you get cross-linking that would also have the opposite effect, where you would
we have cross-linking also with opposite effect. We have some positions where we get UV-induced activation. We found also some positions where we get constitutive activity, which is then reduced with UV lights. So also this we have, but there I did not go into detail now.
Got it. Great. Andy?
Hi, that was wonderful. I noticed that when in the original experiment with the 20 different sites with both BPA and para-azido-Phe, the results were very different for the 2 amino acids. Almost no residue did the same thing, kind of. And I'm wondering, is that generally true for those 2, that basically if one wants to do a cross-linking study, one should always try both of them at every site you care about, because you never know which one's going to do what you want?
We simply tried both. We know that ATSI is not that specific and BPA is more specific, but the disadvantage of BPA is it's quite big. And yeah, with the TM domains and Orai, they are all quite close together. And yeah, it's true. There are several positions we get with ATSI different results than with BPA. And we have to figure out the reason for this still.
Did you look at the phobicity of that region? I know that like Andrew Plested did some work here where he noticed that the region had some kind of conserved, if it was near lipids or if it was in a water reservoir, if which one worked better or not, or I could be wrong about that, but I believe that there was a study on that.
Yeah, there was a study of Plested concerning BPA that it's working better, but in our hands, in particular, incorporation worked for all the positions quite well. So we tested all of them.